Swayam Prabha. Digital India. Educated India.
Hi everyone, welcome again. So, today we are on the last class of the protein module, and here we are going to discuss protein folding, thermodynamics and kinetic parameters of protein folding, and I also include some very interesting but brief description of protein experiment, which will help you understand how experimentally we characterize protein. So, let us start with folding of protein. Protein folding is the physical process by which a protein chain acquires its native three-dimensional structure, a conformation that is usually biologically functional in an expeditious and reproducible manner. So, what I mean here is that protein folding is a process by which a protein sequence make trial and errors to reach into a form where the protein is compact, the protein is functional. It is the physical process by which a polypeptide folds into its characteristic and functional three-dimensional structure from a random structure. Each protein exists as an unfolded polypeptide or random coil when translated from a sequence of mRNA to a linear chain of amino acids. This polypeptide lack any stable three-dimensional structures. So, you could now understand in the last 4 classes what we have understood: the forces, the non-covalent interactions, disulfide bond, Ramachandran plot, torsion angle, all now come into a proper meaning, and we are where we have started our journey from a sequence. To understand function, we need a three-dimensional structure. So, up to now in the course of structural biology, what I was talking about a structure, structure means fold. How this protein fold? In this point, especially people who are really making their introduction to protein, I would help you to perform a virtual experiment. If you guys are blindfolded, if someone fold your eyes and you cannot see, and a bowl is given to you which is full of 20 different color pulse, you have to pick it randomly, and you are given a string, you will put into the string. What you will make? You will make a necklace. That necklace is called the protein sequence or primary structure of the protein. Now, if you take that necklace, make it folded and give a particular shape, that is called the folded protein. Remember, in the last class I was talking about the possibility of innumerable number of proteins. That is what nature manage it. Nature allow those primary sequence to become a protein where they are good enough to make those non-covalent interaction along with the disulfide bond. So, get a meaningful structure shape, so that either they could work as a structural protein like a fibrous protein, or a functional protein like a globular protein. What is behind? We know the forces, but what thermodynamics is talking about here? Let us check. The thermodynamics of folding. Folding of a globular protein is thermodynamically favored process, that is, del G must be negative. You all know the process of spontaneity is del G equal to del H minus T del S, where del G is the free energy change, del H is the enthalpy change, and del S is the change of entropy. Now, so the condition here is del G have to be negative. For del G to be negative, this sum have to be negative, right. So, del H have to be negative, whereas del S have to be, these are the possibilities, right. Let us check them. The folding process involves going from a multitude of random coil conformation to a single folded structure, from a random coil to a fold. The folding process involves a decrease in randomness and thus a decrease in the entropy and an overall positive contribution to del G. This decrease in entropy is called conformational entropy. So, you see what they are talking about. They are saying from this to this process, which is called the folding process. This folding process involves a decrease in randomness. What we mean by randomness, or what we mean by entropy? Let us take a interesting example. There is a stadium. The stadium is centered or focused on a stage. Now, you enter there, the audience applaud. If you are a common man like me, the audience would applaud, but they would not cheer in a frenetic way. And then come Salman Khan. Suddenly the stadium erupted. So, this is entropy. Salman Khan brings more entropy than you or me. So, if this change, then del G would be more negative. So, process would be spontaneous. That is why people pay him, because he make the process spontaneous. Coming back to the protein folding, the folding process involves a decrease in randomness, because there is a coil and it does not know the particular positioning. Now, when it is folded, it get particular fold. So, it is not that random, the entropy is decreased, the whole audience is becoming calmer here. So, this decrease is contributing to a overall positive contribution to del G. This decrease in entropy is called conformational entropy. An overall negative del G, a result of features that yield a large negative del H enthalpy change or some other increase in entropy on folding. So, let us come to enthalpic contribution. Formation of secondary structure is an enthalpy driven process. Why? If you remember, there are so many hydrogen bonds developed. If you think mainly on the main chain, there is CONH for alpha helix, it was N2 N plus 4 for parallel beta sheaths, it is having angles. For anti-parallel, it is straight. So, there are so many hydrogen bonds. So, energy derived from the formation of many van der Waals and hydrogen bonding interaction as well as alignment of dipoles overcomes the loss of entropy associated with the formation of peptide backbone conformation. I was only talking about this hydrogen bond, but because of this alignment, the alpha helix, the dipole formation, all is favoring the enthalpy. Formation of tertiary structure is enthalpically unfavorable. Energy loss in the barring of ion pairs and the breaking of shorter stronger H2O bonds. So, the shorter bonds when they are packing, they are breaking. So, and also the barring of ion pairs, because you have to develop the hydrophobic core. So, the ion pairs are barring there and that results in enthalpic loss. Though some energy is gained from van der Waals packing, the hydrophobic packing, very little is gained from the formation of internal hydrogen bonds, because as many hydrogen bonds with water are broken as are formed in the process of folding a protein. So, suppose there is a alpha helix followed by a beta sheet, another beta sheet. So, they are far away and they are coming together. When they are coming together, they are making layers, the water molecules start being removed. So, as many hydrogen bonds with water are broken as are formed in the process of folding of protein. Free energy associated with solvation of an ion is minus 60 kilocalorie per mole. So, an ion will not be buried in the hydrophobic interior. So, when we talk about the real ion forms, let us say a metal ion, let us say which are forming hydrophilic surface, they are not burying. So, they are contributing to favorable energy. So, protein folding we call entropy driven process. Upon protein folding, the hydrophobic residues move to the interior of the protein, the caged water molecules are released, then enthalpy is gained. Entropy is also gained, which is extremely favorable, because you know this is del G equal to del H minus T del S. So, when this is gained is not good, but when this is gained, this is good, because if it is minus, then it would be contributing positively. Increase in entropy of water compensate for the loss of conformational entropy of the protein and drives the protein folding process. If you remember, I talk about that any macromolecule, the thermodynamic process. In future, you will see I will talk about the process of crystallization, process of protein folding, process of crystallization, which are stabilizing process kind of going through similar thermodynamic pathway. So, coming to the free energy of folding, difference in energy, the free energy between folded, the native and unfolded state is mole 5 to 15 kilocalorie per mole. Enthalpy and entropy difference balance each other, which we talk about, and delta G is a small positive number. So, when we say that the delta G would be small, what that means? It is necessary because too large a free energy change would mean a very stable protein. So, it favors the process of protein folding the difference is not much, because too large a free energy would mean a very stable protein. If the protein is very stable, what will happen? It would not be able to change its conformation frequently, and because of that, as we discussed, when protein have to be functional, it changes it. There are small non-covalent forces like hydrogen bond, hydrophobic interactions which are altered, and there is a clear scenario of change of conformation of protein. But if one protein conformation would be very stable, then the alteration would not happen. Let us say a protein is here, now a substrate is coming, the protein have to be opening up, that is a change of conformation. If a protein is very stable, that change of conformation would not happen. However, structural flexibility is important to protein function, and proteins need that for switching. So, protein folding, as we discussed, from a random sequence to a native conformation, as you see the change is happening here, you see that the protein is attaining the minima. So, what are the forces that guide this process? We discuss what are the steps involved and how fast can this happen. These are question for millions of years, especially last more than 100 years we are doing intense research about this. And one of the breakthrough which I have to talk about is from Christian and Finson in 1972, who told that the native folded structure of a protein under optimal condition is the most energetically stable conformation possible. And most of the information for determining the three-dimensional structure of a protein is carried in its amino acid sequence. Very important. Before this, it was thought that the protein being a biological molecule, the protein's folding depends on the system. So, protein folding is a biological phenomena. But he wants to change the idea, and in his paper Principles that govern the folding of protein chains, published in Science in 1973, he changed the idea. Now, he had performed some experiment, it is called ribonuclease refolding experiment. So, ribonuclease, if you see, is a protein which is having alpha helix and beta sheets. Now, interestingly, the protein have several cysteines, around 8 cysteines. So, the reason Christian and Finson have chosen ribonuclease is not because of this, but mainly because it used to develop a assay where a colorless solution is converted to red color. So, ribonuclease is a small protein that contains 8 cysteine linked via 4 disulfide bonds. As you see here, the 4 disulfide bonds. So, that time you could not measure folding, but the hypothesis was established up to then that if a protein is functional, the protein is folded. So, functional means folded, and the assay I mentioned, and Finsen used it. So, he purified the protein and did the assay and so that the protein is able to generate red color, means the protein is folded. Then he used urea and 2-markaptoethanol, which fully denature the protein. So, from folded he bring it to the unfolded state, and he proves that the protein is unfolded because he performed experiment successful. Experiment failure, it cannot convert the colorless solution to the red color. When urea and 2-markaptoethanol are removed, the protein spontaneously folds and the correct disulfide bonds are reformed. So, he have purified biologically, used the assay, shown the assay is working, protein is folded. Now, he denature, perform the experiment, protein is not working. So, here it is folded, here it is not folded. Then again he removed those denaturing agent and experiment successful, means folded. So, from there what he is talking about, he is talking, see, I had taken the protein from biology, but I have denatured it and then I remove them, but when I remove them the protein renature by itself. That is the key of this experiment. Folded protein taken from biology, unfolded, and now it is folded. So, it had no biology there, it is the solution. So, the sequence alone determines the native conformation. What he is concluding, the sequence, remember how to fold, you do not need to work with or you do not need to help with any biological factors. Without those biological factors the protein could be folded. It is a quite simple experiment, but it was so crucial that he was awarded with Nobel Prize. And if you do not understand, I am again summarizing the fact that before this Christian and Finsen's experiment, nobody worked with protein outside the biological system. If you remember, when I talked about the early days of crystallographic studies or protein studies, most of them are collected from blood because you could get the blood in pure form. So, people thought when you are taking the blood, they have the biological factors together. But now with Christian and Finsen's experiment, it is proved that you do not need other factors, you could individually work on a protein and characterize it purely. That is the change of history. So, remember I talked about the protein could fold biologically the conclusion from Christian and Finson's experiment, protein could fold using physics and chemistry. What I mean? A sequence remembers where to fold, so it fold not by biology, but by remembering the size, charge, distance, interaction. That is why I told it is folding by physics and chemistry instead of biology. This have changed the era, but the situation of that time would not be complete if I do not talk about Levinthal. Levinthal was a mathematician. When Christian and Finson have performed this experiment, critics told that okay, this is successful for probably ribonuclease, this would not be successful for other proteins. Unfortunately, Christian and Finson have no other experimental way to show that, because that time protein folding was not possible to instrumentally or experimentally demonstrate or display. But Christian and Finson got a huge backup from Levinthal. What Levinthal told, assume that there are 3 conformations of each amino acid. So, if this is the amino acid, this is one conformation, you put on right and left, you get 1, 2, 3 conformation. These are the minimum conformation you have to consider for anything. So, consider that minimum number of conformation for each amino acid. Now, if you take a average protein, its size range from 200 to 300 amino acid. So, he had taken the lowest one, a protein with 200 amino acid. So, the total number of conformation is 3 to the power 200 is equal to 26 into 10 to the power 95. Now, he consider if 100 picosecond, which is 10 to the power minus 10 second, is required to convert from one conformation to other, a random search of all conformation would require 26 into 10 to the power 95 into 10 to the power minus 10 second, which is actually 3.2 into 10 to the power 30 years. However, we all know that protein take time of folding from millisecond to second order. So, what Levinthal proved is the protein folding is not a random phenomena. The protein is not randomly searching for a stable interaction and then finding a stable interaction. Rather, it is a sophisticated search process. It is a thermodynamically driven process. That is what Levinthal wants to prove, and together they have changed the spectrum. They have bring what we call today in vitro experiments, like one protein you pick it out, you work on the pure protein and know about it. That is the contribution, that is a change of the way we look at the experiment. I agree, Christian and Finson did a wonderful job, and Christian and Finson deserves a Nobel Prize. But one thing which I have seen student or even us misunderstand about Christian and Finson experiment is that, you know, when we are talking about two situations, one here I talked about in vitro situation. In the in vitro situation, if you see, the protein is in much diluted situation, much safer situation. By safer I mean, here you are maintaining proper temperature, proper buffering condition, and you are making a dilute protein. Whereas, when we look at a in vivo situation, a protein do not have that much facility. Imagine you have to study something and you have to make a write up out of that. Now, it would be very easy for you to study in your study room alone, you get time to concentrate. Instead of sitting in your study room, if someone asks you to go to a busy metro station of Delhi and do the same thing, I am not saying that you would not be able to do that. What I am saying is, you have to get a new mechanism. So, what I mean is, from your study room and from a busy metro station of Delhi, from both of them probably you could do a good job, a good write up. So, the results are same, but the process could not be same. Similarly, what Christian and Finson found in vitro situation, the protein folding. So, the protein folding here in a in vitro situation and in a in vivo situation, the results same, but the journey is not same, because in the in vivo situation, in presence of so many protein, in presence of the fear of proteolysis and lot of other physiological problems, the protein have to get a folding mechanism different, and that is where it might take the help of a chaperone. So, what I am trying to say is, protein folding, the end result is same for in vitro and in vivo situation, but the journey to attain the folded protein is different. It is not true that protein folding is not a biological phenomena. It is still true for the in vivo system, whereas for a in vitro system it is more a physico-chemical phenomena. So, for any given protein, there is one conformation that has significantly lower free energy than any other state. It would be achieved through kinetic pathway of unstable intermediates. So, if you look at, we are talking about, you will see that the protein have a complex energy graph, and from unfolded protein comes to folded, in between there are other states, and molten globule is a established theory which is intermediate. Assisted by chaperones and protein disulfide isomerases, which I was talking about, intermediates are not trapped in local low energy state. So, coming to the kinetic theory of protein folding, folding proceeds through a definite series of steps or a pathway. A protein does not try out all possible rotation of conformational angles, but only enough to find the pathway. The final state may not be the most stable conformation possible, but it could be the most stable conformation that is accessible in a reasonable amount of time. So, what it means that if this is the pathway, probably it is not achieving here, but the energy state is in this region. This is also the biologically important time frame of folding. As I was talking about, this is the energy picture where you see unfolded to the native state. Protein folding is initiated by reversible and rapid formation of local secondary structures, alpha helix and beta sheets. Secondary structure then form domains through the cooperative aggregation of folding nuclei, and domains finally form the final protein through the molten globule intermediates. Molten globules are where the hydrophobic collapse happened. So, it is a hydrophobic moiety, and you will see the hydrophobic moiety is exposed here. So, how you know your protein have a molten globule? Very interesting experiment. If you are putting heat in the protein solution you have, what will happen? The protein will open gradually. You could do that by adding pH also. What will happen? It will break the non-covalent bond of the proteins. So, you have a protein, you provide heat, what you expect is folded state to unfolded state, but sometime it happen that it unfolds through intermediates which are molten globules. So, you see that the hydrophobic domains patches which were buried inside are now exposed. If you use a compound called ANS and perform a fluorescence spectroscopy experiment for a protein which goes through folded to unfolded, you would not see any change, but for a protein which is going through folded to molten globule to unfolded, you will see a change in the spectra. You know that ANS bind there, because if ANS bind to something, its frequency would be restricted and it would show the fluorescence. There are different proposed model for protein folding: hydrophobic collapse, formation of molten globule which I talked about, framework model, secondary structure forms first, perhaps including super secondary structures and then going to the final structure, or nucleation which domains fold independently and subdomains serve as structural kernels to help the protein domains fold. I will only discuss about framework model. Molten globule I talked about, it is the local interaction, they are the main determinants of protein structure. The interactions as the one responsible for forming secondary structural element alpha helices beta sheets, which we talked about numerous time in this module. Isolated helices and sheets form early in the protein folding pathway, then assemble in the native tertiary structures. So, this type of concept, you have unfolded, then you have secondary structures, then you have intermediate compact state, and then you come to the native structure, that is called framework model. So, what is really happening? The hydrophobic side chains are being buried, secondary structure formation insulate the polar protein backbone from the non-polar protein interior. So, it is like you could think in that way, it is like these, and then from these it comes like that, and then these hydrophobics are coming and they are surrounded by, so hydrophobic residues, hydrophobics, and here you get more hydrophilic. Hydrogen bond, disulfide bonds and salt bridges begin to form and stabilize the structure. Van der Waals interactions bring protein substructures into stable contact. What are the most important in folding? non-specific interactions, hydrophobic effect, van der Waals are required to bring the protein together into a globular conformation. Steric interactions, restraint on the backbone, limit the ways in which the globular conformation can form, and if you remember, this is where we are getting our Ramachandran plot. Chemically specific interactions, hydrogen bonds, ionic interactions, salt bridge, dipolar interactions determine the fine detail of the protein structure. There are enzymes which speed up the folding, protein disulfide isomerase facilitate formation of correct disulfide bridges. Peptidyl proline isomerase catalyzes cis trans isomerization of peptide bond involving proline, which we have discussed. Proline, because of its locked structure, it is in the cis form. So, that is one of the obstacle, and last but not the least, molecular chaperones, it helps folding especially of large protein by preventing interaction with other proteins. I want to talk about them, but as I told, this is the introductory course. So, I talked about, I mentioned them, but I did not go into detail, rather I will give you a problem which I will discuss later with lot of experimental evidence. What is the molecular basis of the observation that proteins of thermophiles are more stable than homologues in mesophiles? I explain the question. So, same protein in thermophile. Who are thermophiles? Thermophiles are organism which live in high temperature. For example, if you go to hot spring and you collect bacteria from there, you will definitely find that a protein, a gene product, I would say, coming from that bacteria, if you study, it would show more stability than the same protein in a bacteria which is staying in normal temperature. That is called mesophile. So, why this is happening? Give your thought, we will come to explain that. In the second part of today's lecture, I will give you a very brief introduction to the techniques related to study proteins. I will start with very simple protein quantification method. There are different methods, but I will talk about biuret method, Lowry method, bisynchronic acid or BCA assay, and dye binding assay. I will take example of Bradford assay. So, biuret method is a test for detecting the presence of peptide bond. Under alkaline condition, copper forms a violet colored complex. Protein concentration is directly proportional to the intensity of color absorbed at 540 nanometer. The biuret reagent contains sodium hydroxide, hydrated copper to sulfate, and potassium sodium tartrate to stabilize the complex. Reduction of copper results in a color change. You see the change of color, which can be read at 550 nanometer. The linear range is typically 0.5 to 20 milligram of protein, which means by using biuret method you could estimate the concentration of protein 0.5 milligram per ml to 20 milligram, and you see this is the complex form using the biuret method. Lowry method, it is advanced from biuret. What happened? It is named after Oliver Lowry, who introduced a more sensitive assay. It is as I told, a modification of biuret test. The Lowry method relies on the reaction of copper with protein, the same way biuret used to be done, but the sample is also incubated with a reagent called Folin Thiocal2 reagent. So, you see that this is the complex here. The reduction of the reagent under alkaline condition results in an intense blue color that absorbs at 750 nanometer. The Lowry method is best used with protein concentration 0.01 to 1 mg per ml. Tyrosine, tryptophan and cysteine of protein reduce molybdenum acid and phosphotungstic acid of a phenol reagent, turning the solution blue. The concentration range is 5 to 200 microgram per ml. So, this is estimated with the protein bovine serum albumin. Bicinchoninic acid, it is again similar to Lowry, except here the BCA is used replacing the Folin Thiocal2 reagent. After reduction of copper ions, 2 molecule of BCA chelates with each copper ion, resulting in formation of an intense purple color that absorb at 560 nanometer. This test is as sensitive as Lowry method and works well with the protein concentration from 0.5 microgram per ml to 1.5 milligram per ml. Although detergents do not interfere as strongly as in the Lowry method, other contaminants can interfere. In addition, aromatic amino acids can influence the reaction. However, at higher temperatures, peptide bond can also contribute to the formation of the product. Therefore, for these problems, it is recommended to perform the reaction at higher temperatures to increase the sensitivity and decrease the variability due to amino acid composition. Concentration range 20 to 2000 microgram per ml. So, dye binding assays rely on an absorbance shift that occur when a dye bind to protein. You have a dye, it bind to protein, the absorbance change from the unbound and bound dye. Several different dyes could be used: Comasi Brilliant Blue G250, which we are going to talk about, Bromocresol Green, Pyrogalol Red, but most commonly used dye binding assay is Bradford assay. So, we will read about that. The Bradford assay is an easy, sensitive and accurate method for protein quantification. Binding of Comasi Brilliant Blue G250 to proteins causes a shift of the dye from red 465 to blue 595 nanometer under acidic condition. It is compatible with more common reagents, although detergent can cause interference. Protein with a concentration of 20 to 2000 microgram per ml can be measured using Bradford assay. So, how it is used to measure concentration? Take 200 microliter of the reagent, add 800 microliter. So, when we say this reagent, this is like reagent plus water. So, whenever you get it commercially, you have to make it diluted. So, you have a 1 ml of the solution, add 2 microliter of the protein, change of color is observed and intensity measured at 595. You see that when you keep changing higher concentration of protein, you gradually start getting more intense blue color. And now you compare protein production from different culture. So, you have different culture and you want to know the protein content. So, you pick up the cell, you spin up, so you get the cells and then you add lysis buffer to lyse the cell and then add the solution here. You could also do real quantification. So, for that you have to use BSA as standard. So, you have different concentration of BSA, you put them and you get a curve. Now, for an unknown protein you do the same thing, put the curve and you know the concentration. For BSA you know the concentrations. So, you know where it comes. For the unknown curve you compare with BSA and you know what is the concentration. So, using those reagents you could identify protein, you could quantify protein, you could understand or compare different cultures, you could calculate the concentration of a unknown newly produced protein. Another very interesting technique is gel electrophoresis, and this is a very handy technique. So, what is the principle? You have the protein might have disulfide bond and might have positive or negatively charged surface. So, what you do to reduce the disulfide bond, you add 2 mercaptoethanol and you boil. Also you add sodium dodecyl sulphate, that is why it is called SDS gel electrophoresis. So, when you add mercaptoethanol, it is reduced. If it have the disulfide bond that breaks, then you boil it, so it is denatured, again you add SDS which also help it denature, but more importantly SDS being the negative charge ion, now the entire protein is bound with SDS. So, all of them are negatively charged. Now, what you do, you have a apparatus like that and you load your protein here and you set up the chamber like that, it goes from the negative to positive. If you remember, the protein is negative, now it would go to the positive direction. So, when you run the gel, then you stain it and you will see the bands. Nowadays there are advanced markers which will give bands even without staining. So, you have those molecular marker. If you see here, you have myosin, you have beta galactosidase, you have glycogen, phosphorylase B, bovine serum albumin, ovalbumin, carbonic anhydrase, suabin trypsin inhibitor, lysozyme, all those, and comparing with those you could get the molecular weight of your protein. Also from the relative migration you could get the mass of the unknown protein more accurate. I have already talked about the concept of isoelectric points. So, you remember in isoelectric point is different for a protein whose side chain is not neutral, whose side chain is acidic, whose side chain is basic. That is what written here. Based on those side chains, pK value would be different and Pi would be different. Pi is isoelectric point, I have discussed. It is the pH at which molecule has net 0 charge determined using computer program for known sequence or empirically by having those pKa values. So, we have already discussed about those calculation. Here is the example of isoelectric point of some protein. So, we have discussed isoelectric point of amino acids, but when it come to protein it would be complex. For pepsin you will see this is less than 1, egg albumin 4.6, serum albumin 4.9, urease 5.0, beta lactoglobulin 5.2, hemoglobin 6.8, myoglobin 7, chymotrypsinogen 9.5, cytochrome C 10.7, lysozyme 11. So, these are different PIs of different protein. According to that PIs, now you could perform isoelectric focusing, it is electrophoresis through polyacrylamide gel in which there is a pH gradient. So, you start in time 1 they are random, then gradually they are separated and ultimately they are separated from anode to cathode because of the isoelectric focusing, the difference in the pH gradient along with the different PIs. Now, you combine these two techniques, isoelectric focusing and gel electrophoresis. It is called 2-dimensional gel electrophoresis. The goal of 2-dimensional electrophoresis is to separate and display all gene products present, let us say in a cell you are experimenting. It is the only method currently available which is capable of simultaneously separating thousands of protein. That is the beauty. There are many disadvantages, I am coming, but the advantage is you have a cell and you do this in the first dimension, you do isoelectric focusing, that is you have a pH gradient and according to Pi they are separated. Then you load it into a SDS polyacrylamide gel which I talked earlier. So, now they are differentiated according to mass. So, you will get a very noisy gel like that, but you could pick up things from here and you could perform mass spectrometry based analysis, which I am talking later. Centrifugation is a very important technique, low speed, high speed or ultra centrifugation, different spin speed and g forces. So, it needs different work to do. You have a cell growing, you want to get a pellet. So, you have the lowest one, you have protein to isolate. So, you have higher speed, and then ultra centrifugation you have to differentiate between cell organelles or something. So, centrifugational methods, differential which is pelleting, simple method for pelleting large particle using fixed angle rotor, pellet at bottom of tube versus supernated solution above. So, the pellet would be at the bottom, you will throw the supernatant if you do not need, and you take the pellet, there your cell or what you need. Zonal ultra centrifugation, sucrose gradient, and it would be in a swinging bucket rotor. Equilibrium density gradient ultra centrifugation, which is cesium chloride, swinging bucket or fixed angle rotor, both are validated. So, these are the process. Sucrose density gradient ultra centrifugation is a powerful technique for fractionating macromolecules like DNA, RNA, protein, organelles also. For this purpose, a sample containing a mixture of different size macromolecule is layered on the surface of a gradient whose density increases linearly from top to bottom. If you see, you will see the changes in the level. So, that is it about sucrose density. About density gradient centrifugation, this was developed by Michelson and Stahl in their famous experiment. It employed a process called equilibrium sedimentation or isopenic centrifugation. Equilibrium sedimentation is a process by which particles in a solution reach a point where they reach their isopenic position and stop moving. If you see, they stop moving and then you could isolate it, that is a process of purification. In name of purification, it reminds me there is very famous method of chromatography. I did not discuss because in the crystallography section I will discuss them in detail. So, here it is separated by sedimentation coefficient. If you remember in ribosome, you say 60S, 80S, 40S, 50S, 23S. These S is Wedberg unit. This is sedimentation coefficient. So, how a protein or polypeptide would be cleaved for further analysis? You could use very strong acid like 6 molar HCl, but you would not get any specificity there. You use sequence specific proteolytic enzymes which are called proteases. You could use sequence specific chemical cleavage, that is cyanogen bromide cleavage at methionine residues. Among them, the protease enzymes are most commonly used and they are in diverse. So, if you take a look, trypsin cleavage points are basic amino acids lysine and arginine. Then you get submaxillarous protease which cuts in arginine specifically, chymotrypsin, phenylalanine, tryptophan, tyrosine, Staphylococcus aureus V8 protease, aspartate and glue, the acidic ones, ASP-N protease, aspartate and glutamate, pepsin, phenylalanine, tryptophan, tyrosine, endoproteinase lysine C in lysine, cyanogen bromide in methionine. So, these are all protease and this is chemical. So, how cyanogen bromide cleave the methionine residue? you will see that the cyanogen bromide, it attacks and it clip the residue between the methyl group and the sulfur, and it form a homoserine lactone. It could mainly work on methionine, it could be used in combination with trypsin. Edman degradation is a very famous method. I talked about this earlier. It was developed by Fair Edman, is a method of sequencing amino acid in a peptide. In this method, the amino terminal residue is labeled and clipped from the peptide without disrupting the peptide bonds between other amino acid residues. Phenyl isothiocyanate is reacted with an uncharged N terminal amino group under mildly alkaline condition to form a cyclical phenyl thiocarbamoyl derivative, and it does not affect the other peptides. Then under acidic condition, this derivative of the terminal amino acid is cleaved as a thiazolinone derivative, and ultimately this thiazolinone amino acid is selectively extracted into an organic solvent and treated with acid to form a more stable PTH or phenyl thiohydantoin. This can be identified by using chromatography or electrophoresis. So, in this way you could keep identifying the N terminus amino acid. When you clip them, you could separate them by using high performance liquid chromatography. This is a very popular method of identifying small molecules. Mass spectrometry is a very popular method. Generally, two type of mass spectrometry method is used. Here I am talking about peptide mass fingerprinting. You have to do the proteolytic digestion of protein. For any process you have to do that first. Then you determine the M by Z of peptides by mass spectrometry, either by multi-top or ESI top electrospray. Then you search fingerprint from the database, and the success of this method is depending on the quality and completeness of the database of specific proteome where you are working. Other method is known as tandem mass spectrometry. Here is the same thing, you do the proteolytic digestion of protein, but here you do the same step, but then you do a separation, you could say. So, you could do the proteolytic digestion of protein, instead of then going there for doing M by Z, you do a separation and then determination of M by Z of each fragment. This is called the first step, and then determination of collision induced dissociation fragment spectrum of each of the peptides separately. Because of that, it is high quality and gives context sequence dependent information. It is more applicable to de novo sequencing, where you have no idea or no standard, no database. How you locate the disulfide bond? If you have a disulfide bond present, you either do the reduction by adding dithioerythritol or do the oxidation by performic acid. In the case of performic acid, you get cystic acid residues. In case of dithioerythritol, the disulfide bond would be reduced and individual SH group would be formed, and then you put iodoacetate, it would do acetylation, and now you get acetylated cysteine residues. So, in the protein you identify the acetylated cysteine residues and you know that where the disulfide bonds are there. So, without treating with dithioerythritol, with treating with dithioerythritol, you will get the difference. All the process we talked about, you could apply all of them, and what you do the reduction of disulfide bound using dithioerythritol, as we told, you apply trypsin, the protease which would cut into arginine and lysine, you apply CNBr which would cut into methionine, and then you do Edman degradation to know the N terminus ones, and you apply mass spectrometry to get a fingerprint of the sequence. So, once you are working, as I told about, you have the sample, you do the 2D electrophoresis, you get the pure protein, you do the proteolytic digestion and get the proteolytic fragments. That proteolytic fragment will go through the multi top, you get the mass spectrum, or you purify with capillary electrophoresis, and then you go for LC-MS for amino acid sequencing, where with the mass spectrum or with the fragment you go for database searching. But then you have a unknown protein, you do again protease digestion, you get the fragments P1, P2, P3, P4. With them you go for multi top, these fragments are there, you get M by Z values for each of them, and you know their mass. Now, you search the database, you get the candidate protein, you make the digestion simulation and you calculate the molecular weight, you compare the peptides and identify the unknown proteins. Again, if you want to understand the proteomic modification, you do the same thing, unknown protein, protease digestion, get the fragment, liquid chromatography, isolate or purify the fragments, do the ESI mass and then get the sequencing. So, it is called direct sequencing. For a phosphorylated protein, you do that, you get the fragments, the P4 fragment is phosphorylated, others are not, you do liquid chromatography to purify P4 and then do ESI mass, you get phosphorylation site. So, in that way you could have identified the unknown protein. Thank you very much.